am Choong Jin Yuan, one of the members from Team BIL. Today, I am going to do an experiment, which is chromatography experiment. First of all, we have some materials needed, and it is magic pens, different color magic pens, pens or pencil, oops, 2 clips if you have these clips. If you don't have, you could use tape. And last but not least is a cup of water, plain water. Now let's start with our experiment. First of all, we need to use the pencil or a pen to wrap the paper like this. Then we use the scotch tape to stick together like this. Okay, let's start with our experiment. Remember, the paper must be thin enough. We could choose some of the color to make our experiment. First of all, I use purple. Next, I will use red. Now it's blue. Last but not least, green. Let it dry for a second. Yes, it's dry. Okay, then we use this cup of water. I will put it inside the water. We need to wait some time. Yes, it works. I need to come out with it. Wow, it's beautiful. Yay, it works. It's done! Hey! Look, it's done! Okay guys, this experiment works! Thank you all for looking at my video. Thank you!
Hi everyone, I'm Yien Yhee from Team BIL, also known as Bring Ideas Alive, from SMJK HENG. So today we are going to do the DNA extraction from fruits, so let's get started. So the first step is to mash up the fruit of your choice in a bowl. I choose kiwis for this experiment. So this is the mashed up kiwis. I've already done that step in advance so that this video doesn't take too much time, so this is the kiwi. Okay, so step 2 is in a separate bowl. We need to mix the washing up liquid. Here it is, the 5 gram washing up liquid, and then salt, 2 grams of salt, and 100 ml of tap water. So we mix it together. Then we're going to mix it gently, and you shouldn't stir it too hard to avoid making too many bubbles. So let's stir it. Okay, and yeah, I think we've done the step 2. Step 3 is to add the fruit, the mashed up fruit, to the extraction buffer and mix again. Okay, so let's try to do it quickly. This should be good. So now that we've done step 3, we need to make a water bath with a temperature of about 60 degrees. Okay, so let's put this aside. Here is the water bath. I'm going to pour in the hot water, and here is the hot water. So I'm going to pour it inside this bowl. Oh. Then I'm going to transfer the extraction buffer into this white bowl here. Then we're going to put it inside the water bath. And now we're going to let it to incubate for 15 minutes. So I'll see you guys after 15 minutes. Hi, welcome back. So after 15 minutes, we should take it off the water bath. I'm going to use a wet cloth to protect myself. Okay, let's move this aside. Now we're going to filter it through a sieve. Now we are going to put ice cold alcohol, with this 100 ml, into the extraction. now we're going to see if there is a white cloud-like surface forming. So yeah, I'm just going to wait for that. Okay, so I'm going to attempt to take out the DNA. This should be the DNA from the kiwi extraction. So right now I'm going to put all the DNAs I can from here inside this cup. So I'll show you the end results in a minute. Here's a kiwi DNA. Thank you for watching. Bye!
Hi everyone, my name is Seoh Shan Wen and I am a group members and also a team leaders from Team BILs from SMJK HENG Yhee. And now I'm going to answer the questions for the observation and results from Yien Yhee's performance, which is DNA extractions from fruit. For the first questions, the DNA extraction solutions contains washing up liquid, which is a kind of a detergent, and detergents are very good at dissolving away fatty molecules. Just like when you are washing the greasy dinner plates after a tasty roast lunch, while salt helps to dissolve the DNA into the solutions. For hot water, it helps to soften the cell walls and membranes, so the DNA is released. We need to use alcohol because DNA does not dissolve very well in the cold or concentrated alcohol, so when the DNA molecules drift up from the fruit juice into the alcohol, they cannot longer stay dissolved. And for the second question, when you added the salt and detergent mixture to the smashed fruits, the detergent helps lyse the fruit cells, release the DNA into solutions, whereas the salt helps create an environment where the different DNA strands could gather and clump, making it easier for you to see them. After you added the cold rubbing alcohol to the filtered fruit liquid, the alcohol should have precipitated the DNA out of the liquid while the rest of the liquid remained in solutions. You should have seen the white or clear gooey DNA strands in the alcohol layer, as well as between the two layers. A single strand of DNA is extremely thin, too thin to see the naked eyes. But because the DNA clumped in this activity, you are able to see how much of three fruits have when all their octoploid cells are combined. For the last questions, I think that the sample that I have prepared is not pure DNA because the fruit cells contains membranes and proteins. If we want to get a pure DNA, we must separate and remove these unwanted materials first. Thank you for listening. Bye!